All right, so when we look at nucleotides, one of the more important things about it, or about them, is the nomenclature. So we have nucleic acids and we have nucleotides. Our foundation is a base. That base is going to be either a purine or a pyrimidine. Now, when that base is combined with a sugar, you make a nucleoside. Nucleosides have unique names based on the base that is present. So adenine plus a ribose sugar makes adenosine. Now, the nucleotide version of that also has a phosphate. So adenine, ribose, and at least one phosphate group, you're going to make the molecule known as adenylate. Okay, so we have these unique names, adenylate versus deoxyadenylate. The difference there is the sugar that's involved. That is either a deoxy sugar, so deoxyribose, which is found in DNA, compared to ribose. So we've got this kind of hierarchy of naming. So it's important to know if you're talking about a nucleotide, if you're talking about a nucleoside, or if you're talking about a single base. Now, what's common is that you will see or hear people say the name of the nucleoside, I'm just going to say plus phosphate. For instance, adenosine triphosphate, well, that's a nucleoside plus 3 phosphate groups. Now, looking at the structure of our deoxynucleotides, well, here we have the name of our deoxynucleosides, so deoxyadenosine, deoxyguanosine, deoxythymidine, and deoxycytidine. So that is the nitrogenous base, the deoxyribose, but no sugar or no phosphate is present. Now, that is going to be indicated by the lowercase d, indicating our deoxy sugar, and the capital A. DA indicates you're talking about a nucleoside. DADP, for instance, that's deoxyadenosine diphosphate. So that is the nucleoside plus 2 phosphate groups. So that's where this variation comes in with respect to a symbol. So capital A is indicating simply the nitrogenous base. DA indicates the deoxy sugar plus the nitrogenous base. DAMP is that deoxyribose adenosine monophosphate. Now, ribonucleotides. So when you have the oxygenated form of that sugar, which I didn't really emphasize that in the previous slide, but I want your attention to be drawn to that carbon right there. That's the carbon number 2'. And on the previous slide, that 2' carbon did not have that OH group. Okay, so we've got our nitrogenous base, we've got our ribose, as opposed to deoxyribose. Our symbols are A for just the nitrogenous base and AMP for adenosine monophosphate. Okay, so that's where we're going to have a little bit of a differentiation. That lowercase d indicates deoxy, whereas the capital, the lack of it indicates that you've got the oxygenated form. So it's important that you are able to identify these structures and which sugar or which molecule that you're actually talking about. So I would recognize and I would be comfortable with deoxyadenosine monophosphate, adenosine triphosphate. Those are important to appreciate. Okay, so a beta N-glycosidic linkage or a beta N-glycosidic bond. This is an important bond because it's the bond that exists between the pentose ring and our nitrogenous base, an N-glycosidic bond. I'll also refer to these as an N-glycosidic linkage, effectively the same thing. Now, the bond is formed to the anomeric carbon, that new chiral center that we found whenever we cyclized our sugar. This sugar is going to be in the beta configuration. Now, if you think about that compared to the alpha configuration, that would be a sterically hindered version of it. The bond is formed between N1 on a pyrimidine and N9 in a purine. pyrimidines, remember, are 6-membered rings, whereas purines, I'm sorry, are, there we go, 9-membered bicyclic molecules. The bond is stable toward hydrolysis, especially in pyrimidines. Bond cleavage is going to be, however, catalyzed by a strong acidic condition. So one thing that I previously mentioned was how these nitrogenous bases absorb light at about, or within a range from 250 to 270 nanometers. And here what we have is, using the Lambert-Beer law, A equals epsilon LC, that epsilon is the extinction coefficient, and this gives you a means to correlate the concentration of any of these molecules, AMP, GMP, UMP, DTMP, and CMP, to light absorbed from a light source of 260 nanometers. So this is just to kind of be like, proof is in the pudding of, well, we see that there is actually absorbance at 260 nanometers from these molecules. Now, minor nucleosides, keep in mind, that's a base plus sugar in DNA, can result from modification after DNA synthesis. One example is 5-methocytosine, is common in eukaryotes and found in bacteria as well. N6-methyladenosine is common in bacteria but not found in eukaryotes. So these are both, where is that modification taking place? On the adenosine, which, what is adenosine? That's a purine, so 2 rings. And cytosine, that is a pyrimidine. Our epigenetic marker, this is a way to mark your own DNA so that cells can degrade foreign DNA. So this is kind of it's almost like a very basic immune system of sorts. These are ways to mark genes which should be active in eukaryotes, so these genes can be expressed. And ultimately, these are things that are somewhat responsive to the environment. So if a gene is to be activated, well then that enzyme that's involved in methylating a cytosine at position number 5, or methylating a adenosine at the N6 position, well, that would be activated and does this. Now, for what that actually looks like, here is the N6 methyladenosine. Remember our nitrogen, or our position number 6 there. We've got methylation at position number 5, and then we have an N-methylguanosine at position number 2, and a hydroxymethylcytidine at position number 5. Minor nucleosides in RNA, well, inosine is sometimes found in the wobble position of the anticodon for tRNA. Now, this is made by deaminating adenosine, and it provides ultimately for a more robust and a richer genetic code. Now, that wobble position is in reference to the 3rd nucleotide, NT, in a codon. So XX, this right here is the wobble position. Pseudouridine is found widely in tRNA and rRNA. It's more common in eukaryotes, but found also in eubacteria. It's made from uridine by an isomerization after RNA synthesis. And what this ultimately can do is stabilize the structure of tRNA, and it may also be helpful in the folding of ribosomal RNA. So what that looks like are inosine, or inosine, pseudouridine, 7-methylguanosine, methylation at this position right here, and then 4-thiouridine, which is a thiol ester group right there. Generally, whenever you talk about something like DNA or RNA, you're talking about polynucleotides. So when it comes to polynucleotides, covalent bonds are formed through what are known as phosphodiester linkages. And this well, think about that. Phosphoric acid, for instance. That is, well, it's H3PO4. Now, it's that phosphate ultimately that gives your something like DNA an overall negative charge. It's that negatively charged backbone, or it's that phosphate that gives the DNA molecule a negative charge to it. Now that DNA backbone is generally going to be very stable, which is why we can recover things like dino DNA, but also DNA from mammoths. Hydrolysis is something that does happen. However, it's going to happen at a much faster rate in the presence of an enzyme like DNase. Now, an RNA backbone is, by contrast, much less stable. You still have those phosphate linkages, or those phosphodiester linkages. However, it's much more susceptible to hydrolysis in an aqueous environment. Now, polynucleotides are generally going to form linear polymers, and the directionality of it is going to be read from the 5' end to the 3' end. And so whenever we're talking about a sequence of DNA, we're talking about it from 5' to 3' position or direction. Now, the hydrolysis of RNA, like I said, is generally going to be, it's generally going to take place, but RNA is unstable under alkaline or basic conditions. Now, hydrolysis is also catalyzed by enzymes known as RNases, and RNase enzymes are all over the place. sRNase, as an example, in plants prevents inbreeding. RNase P is a ribozyme, an enzyme that's made up of RNA that processes tRNA precursors. Dicer is an enzyme that cleaves double-stranded RNA into oligonucleotides. It's for protection from viral genomes, because if you have an RNA molecule, an RNA virus comes in, this dicer can potentially detect it. But in more recent years, this dicer has actually been utilized for RNAi to basically knock down a gene. So you can look at an organism when a gene isn't expressed. How does it behave? What does it do? So the mechanism for this hydrolysis would look something like this. So you've got an RNA molecule right here, 5' down to the 3'. Base number 1, base number 2. What happens is a hydroxide ion comes in and picks up this hydrogen, which these electrons are then able to go down towards that phosphorus. And that phosphorus, then the electrons that make up that bond are able to move down towards that oxygen. They're able to pick a hydrogen up, and now all of a sudden you have a resulting cyclic monophosphate derivative. Water is released, and boom, you've got a mixture of 2' and 3' monophosphate derivatives that are resulting, and you've got a shortened RNA molecule. So if we were to look at a DNA molecule and an RNA molecule, what makes them different? Or DNA polynucleotide versus an RNA polynucleotide, what makes them different? Well, a couple of things. What makes them similar to one another is the direction in which they're read, 5' to 3' end. Okay. In addition to that, a phosphodiester linkage. They're basically the exact same. What makes them different from one another is this 2' position. In a DNA molecule, you have simply a hydrogen, whereas in an RNA molecule, you've got an OH group. So hydrogen, OH, hydrogen, OH. The bases that attach to that anomeric carbon, that 1' position, is we've got ATG. By contrast, in an RNA molecule, you're going to have something like uracil that would otherwise take the place of T. So hydrogen bonding that takes place between strands of an RNA molecule, two bases can bond together to form a base pair. Now, those monomers, a large number of base pairs is possible. So in a polynucleotide, only a few exist, or a few possibilities exist. And that's because you're going to have this spatial limitation that's imposed by that polynucleotide structure. So what this means is if you had free DA, well, that could maybe form a hydrogen bond with DT, DG, DC. But whenever we talk about it in a polymer, well, then you have some limitations. So then DA is going to pair with DT within this polynucleotide structure. DG is going to pair with DC. And that's the Watson-Crick base pairing that kind of predominates. So A pairs with T, C pairs with G. What that also amounts to is a purine pairs up with a pyrimidine in a polynucleotide structure. So to visualize that, here we've got adenine and thymine. Here's this larger of the two rings in our adenine molecule, and here are the corresponding places in which a hydrogen bond is formed between A and T. Okay. By contrast, guanine and cytosine. Guanine and cytosine, we've got a total of 3 bonds, whereas in adenine and thymine, you've got 2 bonds. Okay, so all of these bonds are no more than 3 angstroms. Ultimately, what this means is, if you think about it in terms of being held together tightly, well, 2 bonds versus 3 bonds, what's going to have greater force holding them together? When there are 2 almost identical bonds versus 3 almost identical bonds, yeah, those 3 bonds are going to give a more stable structure than the 2 bonds. This image right here was devised by, or elucidated by Rosalind Franklin, who was a key figure in the determination and development of the theory of the double helix of DNA. Watson and Crick are, along with Maurice Wilkins, shared the Nobel Prize in 1962. Rosalind Franklin died in 1958, so she couldn't really receive the acclaim that she's very, very well deserved. But ultimately, they came up with this theory of the double helix where you had 2 base pairs, A and T, that paired up with G and C. And they came up with this model that ultimately is what we still use today. Here's Rosalind Franklin and Maurice Wilkins. They're 2 of the figures that are not as well recognized as Jim Watson and Francis Crick, but they're 2 key figures in development of this double helix. Now, the Watson and Crick model of bDNA is the predominant form of DNA, and here are some of the features about it. First of all, the structural features known as a major and minor groove. These are, and it's tough to tell from a 2-dimensional image, but these are basically spaces within a double helix in which something like a protein can gain access to. Now, if you look here on this space-filled model, you might get a better glance of that major groove, which is right there, compared to the minor groove right there. So this is bDNA, and these are kind of the features of, or spatial features of it. Now, what this corresponds to right here, this 10 angstroms, this is the height of 1 full rotation. And there are approximately 10 nucleotides in that full rotation. 1 base pair, or this distance between these 2 base pairs, is approximately 3.4 angstroms. So it takes 10 angstroms to get 10 nucleotides to get 1 full rotation about that double helix. The width of this double helix is approximately 20 angstroms. So The space between base pairs is approximately the height of one base pair is approximately 3.4 angstroms. One full rotation is going to be, so I'm thinking about it like starting on this end, follow this around, one full rotation is achieved after it's traveled a height of 36 angstroms. Now, B form, like I said, is the predominant form. There are other forms of DNA that can be produced, A form and Z form. Those are much less common, but they are forms that DNA can take. Some differences between those forms is the diameter. For instance, A form is about 26 angstroms long. Z, or wide, is 18 angstroms long. There are about 10.5 angstroms per one full helical turn. A and Z have 11 and 12 respectively. Each base pair in A form versus Z form, well, they're 2.6 and 3.7 angstroms respectively. So that B form is not as compacted as A form, but it's more compacted than Z form. So those are your three major forms of a DNA molecule that can exist. Now, what's most important with respect to DNA? I would argue what's most important with respect to DNA is that your strands are complementary and they run anti-parallel to one another. So one strand is read 5' to 3', the other strand complements it by being in the 3' to 5' direction. But keep in mind that we read it in the 5' to 3' direction. All right, well, this last little bit is something that we talked about in the very first section of the course, that we have this model in which DNA is replicated. Each of these strands provides or acts as the template for the production of a new strand. And that's something that Watson and Crick reported in their 1953 paper. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So pretty profound thoughts there.